Okay, so I wanted to just briefly introduce the AMBON project, which stands for Arctic Marine Biodiversity Observing Network. And so I've listed all the co-PIs on the first slide there, so you know sort of who is involved as well. And this is the result of a competition that was out in 2014 and got funded through the National Ocean Partnership Program. And so there were three so-called demonstration projects funded to look at how can a marine biodiversity observing network maybe function and what could it do. So this is one of the three, and it's located in the Arctic in the Chukchi Sea. And if you would go to the next slide, please. So it is called an end-to-end project, meaning, and that's our slogan also, from microbes to whales. You see that at the bottom. And so the idea is really to cover all aspects of biodiversity, and one of the really new things in all this is really the microbial world, because most ongoing and past projects haven't really focused much on this, but we're also continuing, obviously, with the more known and common groups such as zooplankton, phytoplankton, benthic components, fish, marine mammals, and birds. We do also incorporate biodiversity observations with environmental collections to sort of figure out if there are relationships among those different biota, as well as of the biota with the environment. And so one of the things we wanted to do was to build on existing knowledge and trying to include previous projects and build on those, as well as collaborate with other ongoing projects, and then trying to continue time series that have been started through other projects, continuing those, and then also to close current gaps in taxonomy, and already mentioned microbes as one of those gaps. And then the idea is it's a 5-year project, is that at the end of the 5 years that we would understand better how we can organize a biodiversity observing network for the Arctic as well as for other regions, and for that we work with these other MBONs together that are located in Florida and Southern California. If you go to the next slide, please. I just wanted to introduce briefly where the AMBON sampling region is. So it's in the Chukchi Sea, and you see these little red dots everywhere. Those were sampling stations we covered in our first field effort in 2015. And I have, in light red, I've put some programs there that we are collaborating with, or that we took into account from previous existing knowledge. So you see the DBO, the Distributed Biological Observatory. So we cover 2 lines of the DBO. We're building on the Rusalka project, that is the Russian-American long-term census of the Arctic. And then in the north, we also build on previous occupations, the CSES program and the COMIDA program up there. And then maybe also right above the word COMIDA, you see a little yellow asterisk or star right south of Hannah Shoal, and that is Seth Danielson's Chukchi Sea ecosystem mooring, which is in part also funded by NPRB. So we're right close there, and that is a great long-term record to our more one-time go there sampling per year, and so it's very important for us to integrate with some of these longer-term records of moorings that are out there. You go to the next slide. So this is really just briefly saying what we've been doing in 2015, and I don't want to hold up, I think the call is only supposed to go to 11, I believe. So I'm not going to go into any kind of results here, just showing that in 2015 we've done hydrography sampling. That you see the sampling grid here covered by sea surface temperature. Then we did various chemistry measurements. You see water chlorophyll. We also have sediment chlorophyll, nutrients. Then the next slide is a microbe diversity plot that would be, oops, too far. Can you go back one slide? Thank you. When I said next slide, I'm sorry, I meant the next plot. So the microbes there, darker colors, you can't read obviously the axes, but several microbial groups occur more common in various regions. Zooplankton biomass, we have not completed all the analyses yet. Then benthic and fish species richness with the more reddish colors, higher diversity hotspots versus the blue colors, low diversity. And then this is for seabirds, just the example of auklets, various auklet species, their distribution over the sampling grid, and then also marine mammal observations. And now you can go to the next slide, please. So it's important for us in terms of being a network more than just a in-the-field program, is to really collaborate with other Arctic field programs. And so we are looking forward to talk to Danielle and more about the various programs that have been funded by MPRB, for example, and see how we can, collaborate there. We want to do this through efforts in the field, collaborating and maybe leveraging off each other, but then also through joining data streams. And so we're working with AOS, the Alaska Ocean Observing System, for data integration, and so do many other field projects in the Arctic. And so we are working with AOS on making this more applicable to a greater network. We also have various international Arctic partners in the CBMP. It's the Circumpolar Biodiversity Monitoring Program through the Arctic Council, PAC, ESAS, so there are various partners we have that are Arctic-wide. And then we are also coordinating with the other two MBONs, one funded through in the Florida Keys and Monterey Bay Sanctuaries. And so they work mostly in coral reefs and kelp forests, and then the Channel Islands MBON also working in kelp forests. And you can imagine that trying to figure out commonalities of a biodiversity observing networks in such different systems isn't easy, but we're making quite good progress on that. And so we're working together towards some ideas of a national operational marine biodiversity observing network. We're focusing on essential biodiversity variables for this. So what are those, and how can they be measured, and how can we, be common in our approaches? So there's a lot of talk looking at common methodology, particularly using genomics and eDNA, environmental DNA techniques. And then lastly, we are also part of GeoBon, part of the Working Group 5 on biodiversity observing networks, and we're working with OBIS as another venue for our data to be presented to the public. And with that, I'm actually done in terms of just giving a very brief introduction. Wanted to thank, obviously, the funding agencies. So this particular project is funded through NOAA, BOEM, and Shell. Most of you probably know that Shell has pulled out of their operations in the Arctic for the time being. With that, we actually have experienced some fairly significant loss of funding towards this program. So we're currently working on trying to find ways to deal with this. One of the repercussions of this was to cut our field season in 2016, so this year, but we are planning on going out again in 2017, and hope to make that work. And so with that, I'm happy to answer any questions.
Any questions for Katrin?
Well, this is Ann Jensen, and I do actually have a question.
Sure.
So have you thought about extending your time series backward in time? I'm an archaeologist. I'm a zooarchaeologist. I work with the faunal remains from archaeological sites, some of which are conveniently located near some of your lines, and some of which have several thousand years worth of mostly macrofauna, which you can do all kinds of cool things that are really well preserved. We have two issues with that, one being that social science funding is not the kind of funding you guys enjoy, and since some of these questions are purely or almost purely natural science, it's pretty hard to get funded to to look at it within a social science budget. The second is that these sites are melting, eroding, and whatever, and we will lose all of this data within the next 15 years.
Yeah. We haven't actually really seriously discussed going this far back in time. We have definitely talked about accessing available data that sometimes sit on a shelf in a handwritten notebook somewhere from early projects. But you bring up a really good point, and I think that's definitely something we could maybe talk a little bit more about. I think that's a great idea, because particularly thinking that the benthic environment, I mean, the Chukchi Sea is very much a benthic-driven system, and we just heard it in the presentation before. Some of these higher trophic levels are benthic feeding, such as walrus. The Hannah Shoal area, right south of where we are sampling, is a hotspot for walrus, and we did see a lot of walrus on our lines up there. So having particularly the macrofauna remains is definitely a great tool.
Yeah, I'm actually involved in a large project that's looking at walrus from modern back to archaeological, and we're not anywhere near done with it, but even what we've got so far suggests that they do over time change trophic level and feeding area and various other things. I mean, what you see in the last 30, 40 years is not what they've always done. So,
yeah, correct. We have some collaborators here at UAF who also look at historic samples, basically through bone and teeth records, and it's looking.
It's probably the same project.
That could be.
The same project. So yeah, but I mean the thing is that it's definitely showing that stuff changes through time, and if you're trying to manage them, that would be useful information to have, to say the least.
Absolutely.
Thank you.
Anyway,
are there any other questions for Katrin before we adjourn?
Yes, Jessica Rohde, relaying a question from Natalie Silver. She would like to know if this is different, how this is different from the OER grant.
Yeah. So what Natalie is referring to is what I call the Chukchi Borderlands project, and I actually do have a presentation ready for this, but Danielle and I decided that today there wasn't enough time for both. So this summer we will actually be going up into the Arctic, but north of the AMBON study region, and I'm not sure if it's on the map visible. If you let me just see. No, it's not really visible on the sampling map that I have there. I thought so. It basically goes north of this region where the Chukchi Plateau reaches out into the Arctic Ocean and builds what we call the Chukchi Borderlands. And so this summer, from June 2nd to August 10th, we will be up there with Coast Guard Karahili, and we will do plankton, seafloor, and sea ice work using traditional methods as well as an ROV. And that region is of particular interest because it is quite unknown, but it's bathymetrically and hydrographically very complex. And it is also very much exposed to climate change effects, both through the receding sea ice as well as a very strong influx of warm Atlantic water that is coming from the bottom and is shoaling up in that region up onto, it's not shelf, but onto the plateau there. And so we will be looking at various components again, and we'll be also do collections and do microbial analyses for species identifications there. And so that is quite different from the study region. It's basically like a northernmost or a northern extension of the AMBON region, although some of the ideas of looking at biodiversity and as a metric of how diverse, how rich also in functional patterns is a region is also similar to what we do in AMBON.
Thank you.
Thank you, Katrin. Any other questions? I have a question, Katrin. You mentioned that there is some level of interaction with the other marine bond projects in Florida and California.
Yes.
How is that interaction? For instance, is that retrospective in the sense that you look back at what worked or didn't work, or you try to split different approaches or methods looking towards the future, or both, or how is that interaction? Is it regular?
So basically, what we were tasked with is to see if we can find like common metrics that would be applicable for more or less any marine system, so that if a different region within the U.S., let's say, says we need a biodiversity observing network here, I'm just making this up now, let's say in I don't know, in Maine on the Atlantic shelf there, then what metrics would be the most usable and applicable? And so that is mostly what we're working on, where I mentioned that we're looking at essential biodiversity variables. What are those and what are applicable? And some are obviously more towards some particular habitats than others. And so we're not looking retrospectively so much, although of course we all bring in our knowledge from the past, but we're actually more looking forward into what can be used now in terms of methodology. We're looking very strongly into eDNA applications and trying to standardize methods for these applications. Another thing where we're very strongly collaborating is in data management. So it's one thing to collect the data, but then data management and making data available in a usable format is a very important part of being such a network.
Thank you. Any other questions for Katrin? If not, Danielle, do you have anything else on your end?
I just wanted to say that I think this call was a really nice example of what these IARPC calls are meant to do. We're having conversations like between Katrin and Anne about tying some very disparate fields together to go further. And I think this was really nice. I hope everybody feels like you got something useful out of this month's call. I also just wanted to take an opportunity to mention, when I talked about the NPRB program, I mentioned we'd be having our kickoff meeting next month. If those of you on the phone have ongoing projects that you'd like to explore connecting to what this new integrated program is doing, we would love to talk with you. We're being pretty inclusive in terms of trying to play a coordinating role. I think now that we know the scope of our program and we realize that we're covering springtime observations with input at Bering Strait all the way through the entire U.S. Czech Sea shelf with inputs then into the basin, I think there's a lot of opportunity there to connect with other programs that are ongoing, such as AMBON and others, and AMBON is actually already sort of tied in with what the UAF needs. So, and the other thing I wanted to mention real quickly for the program managers on the phone is if you're interested in looking at other opportunities to connect with us, please contact me. One of the things that we weren't able to find the money to fund directly through this program was the modeling, and we had some really great modeling proposals come in. So there's ample opportunity there to augment the program. So I look forward to continuing our conversations.
Thanks. Any other last-minute comments, questions?